Good afternoon, everyone. My name is Yingxian. This summer, I work at Daniel Kitty's lab, part of Structural Biology Initiative at ASRC, under mentorship of his PhD student, Thomas Geitz, and I will present my summer research project called Virtual Screening to Identify Allosteric Inhibitors Targeting L16 Sites of PDP1B. So, what is PDP1B? PDP1B stands for protein tyrosine phosphatase 1B, which is a negative regulator of the insulin signaling pathway and is a potential therapeutical target of type 2 diabetes. However, because active site inhibitors of PDP1B are non-specific binders, that means they can bind to other proteins rather than PDP1B specifically. That is why we are looking for allosteric inhibitors that can bind to the allosteric sites of PDP1B, which indirectly alter the active site's conformation, and therefore inhibits PDP1B. In previous studies, we found that the loop 16 site is a novel allosteric site available for inhibitor binding, as you can see in the figure here, which comes from a crystallography experiment. There are some experimentally characterized small molecule fragments bound to the L16 site, but these did not show the inhibitory function due to their small size and weakly binding. The hypothesis of this project is to design potential allosteric inhibitors by merging key features of experimentally characterized small molecule fragments in order to increase the number of interactions between ligands and the protein and therefore increase the binding affinity. Methods. First, this is one of the examples to show how we identify the key feature of the small molecule fragment. There is a polar interaction between the carboxyl group of the ligand on the right side of the image and the hydrophobic interaction between either carbon or aromatic ring of the ligand and the hydrophobic pocket of PDP1B shown on the left. Second, the hypothetical merged molecules were designed based on the key features. The figure shows here was one of the merged molecules I designed. It contains a carboxyl group and makes a two adjacent ring structure to enhance the hydrophobicity of the ligand. This is just one of the examples of the merged molecules. Indeed, I designed multiple of these merged molecules based on different key features of the fragments to obtain hundreds of small molecule candidates. Third, a screening library of the small molecule candidates shown here was constructed by searching National Cancer Institute (NCI) database to find the candidates which are similar to the merged molecules. Fourth, 849 small molecule candidates from the screening library were virtually docked to PDP1B 10591 receptor to evaluate their binding affinities to PDP1B protein using AutoDock Winner on CUNY High Performance Computing Center. Finally, after the results were analyzed, the 40 molecules with the most promising predicted binding affinities were identified and visualized in PyMol. Results. The binding affinity scores of 849 small molecules are in the range between -8.2 to -3.0 kilocalories per mole. The 40 molecules having the most negative binding affinity scores were selected, and their scores are in the range between -8.2 to -7.0 kilocalories per mole. The molecule shown in the video is one of the small molecule candidates similar to the merged molecule that I mentioned in the previous slide. It represents ligand-receptor binding interaction in predicted model. The carboxyl group bound to the glutamic acid at the N-terminus of PDP1B. The double aromatic rings in these structures enhance hydrophobic interaction between the molecule and the receptor. Also, another polar interaction between the polar hydrogen and aspartic acid 236 was discovered, which enhanced the binding affinity. In conclusion, designing the hypothetical molecules by merging multiple key features of the small molecule fragments associated with binding interaction helped to identify the most promising predicted molecules which may have inhibitory function. To test if this is the case, these 40 molecules will be ordered from NCI database to study their allosteric effects on PDP-1B in vitro. Also, as we only dock the ligands to one receptor due to time limitation, in future study, more small molecule candidates will be virtually docked to the PDP-1B receptors, which have different conformation. First, I want to express my gratitude to my mentor Tamar Sky and to the PI of my lab, Dr. Daniel Kiddy. They both are principal contributors to the success of my little project. Thanks for all CUNY Summer Research Program coordinator, mentors, and participants for amazing atmosphere and chance to do science even in such desperate time as we are living through right now. These are the references of these projects. Thanks for your listening. Feel free to ask me if you have any questions.